Lecture 4 of Medical Physics 671, Human Physiology and Anatomy. Let's continue our discussion about the physiology of excitable cells. We'll cover the neuromuscular junction and excitation-contraction coupling in this lecture. I'm going to present section 3.6 before section 3.5 in the textbook. Because we've already talked about the resting state potential and the action potential and how that propagates down the axon of the motor neuron. And I think section 3.6 then continues this story by discussing how the motor neuron interfaces with the muscle. And then section 3.5 will talk about the muscle itself. All right, so as a very macroscopic view, we have an axon of the motor neuron that approaches a striated muscle and innervates into the striated muscle. There's 5 to 20,000 muscle fibers that one motor neuron will activate, and that helps with coordination of muscle contraction. Now these termini that innervate into the muscle tissue are called boutons or buttons. I like the word boutons. I think I got its name because on a histology slide, those connections look like little buttons that are sewn onto the muscle. And so you have neurotransmitters that alter ion conductance. There can be other kind of metabolic neurotransmitters as well. And that will change the excitatory postsynaptic potential, increase the potential and activate the muscle. Or there could be inhibitory potentials as well that deactivate the muscle. And I think that's shown quite well here, where you might have up to 10,000 inputs into this motor neuron here in the dendrites and soma of the cell. And some of these could be excitatory, A, B, and C, shown in this graph, terminal A, terminal B, terminal C. But terminal D is actually inhibitory. Well, if I add up the potentials of A, B, and C, well, that's shown here, and you'll fire off an action potential because it got above the action potential threshold for contracting the muscle. However, D could reduce the sum of those action potentials so that's below the threshold, counteracting the excitation caused by sensors A, B, and C. Also, you can have temporal summation as shown in this left column, where terminal A could be stimulated multiple times, and eventually that'll build up. We talked about tetany in a previous lecture. Well, here's an example of that getting above the threshold. The axon hillock is this exposed area that has a lot of sodium receptors in this area that can interact with the extracellular space where sodium is high. And so that can help to depolarize the axon membrane at this location. And if you recall, the depolarization then propagates in both directions along the nerve. However, if you have this one part on one side, well, clearly you're just going to propagate down to the axon itself. Here's really the most important diagram for this section. So again, you have the axon terminal coming into this bouton, and you have these innervations of the membrane, where the membrane of the axon is going into these little fingers that are created within the muscle cell. So you have a lot of surface area interacting between the nerve axon and the muscle cell. And let's zoom into one of these areas here. And we'll just go very straightforward through all 8 of these steps, right? So the first step is for the action potential to propagate down the nerve to the terminal. So you have this depolarization. And that causes a change to this protein, a calcium transporter, the VDCC, voltage-dependent calcium transporter. And that allows calcium then to diffuse from its high concentration outside the cell to the lower concentration within the nerve cell. Well, when that happens, calcium binds to the proteins that dock these vesicles to the outer membrane, the plasma membrane of the nerve cell. And vesicles then integrate within the plasma membrane and open up, spill its contents into this extracellular space between the nerve and muscle membrane. Well, the material inside here is acetylcholine. Acetylcholine is the signaling molecule that interacts between the nerves and the muscle. So that diffuses across that space, binds to the acetylcholine receptor, and that allows the receptor to open up and allow sodium inside the cell. So you had intracellularization of sodium in the nerve, and now you've got intracellularization of sodium in the muscle. Well, then that depolarizes the muscle membrane, and that can be propagated down into the muscle cell further. And then this calcium activation has to be shut off, otherwise the nerve would be constantly stimulating the muscle. So the transporter for calcium closes. Calcium is either bound by intracellular proteins, or importantly, it's pumped back out of the cell so you can fire off the nerve again later on. And that is an active transporter because you're pumping the calcium from low concentration to high concentration. So you need ATP to do that. Also, acetylcholine esterase is an enzyme in this extracellular space that converts acetylcholine to choline and acetate, removes the acetylcholine from the system so that your muscles are not constantly having an influx of sodium. Now we've got to turn off these membrane transporters, of course. Choline is recycled back into the axon. That's important to maintain a high concentration of choline. The acetate is a byproduct that is waste that can go away. So now that we have depolarization of the muscle membrane, that gets transported along the transverse or T-tubule that descends deeply into the muscle cell. That opens up a receptor that then interacts with the sarcoplasmic reticulum within the muscle cell. And we're going to see the SR appear in the next lecture as well. So it's important to get a feeling for how the sarcoplasmic reticulum works. Well, the rhyodine receptor is opened now with this activation of the receptor that's on the muscle plasma membrane. And that allows calcium, which is high concentration within the sarcoplasmic reticulum, a way of storing calcium. It allows the calcium to diffuse into the cytoplasm where it is lower in concentration. And as we'll see soon, that will activate the muscle tissue. Also, the calcium transport is shut off by uptake back into the sarcoplasmic reticulum by SERCA, a membrane transport protein. Also, mitochondria can take up calcium, so calcium is used for multiple reasons in the cell. Okay, so what happens when this calcium reaches the myofilament? Well, first, you have calcium binding to one of the troponin proteins, in particular troponin C. That causes it to modify the conformation of troponin T and troponin I to other troponin proteins. And that tugs on tropomyosin and pulls it away from one part of the F-actin filament. As shown here, you can see that this filament has moved a little bit from its lower orientation on the left to more of a higher orientation on the right. Well, that exposes the binding sites on F-actin to the myosin head group. The myosin head group can then bind to actin and hold the muscle tightly. And we'll talk about how myosin and actin interact very soon. But you need to have this masking effect of tropomyosin removed so that the myosin can bind to the actin, and that's all controlled by the calcium. In fact, this is a cooperative event, so that if you have a low amount of calcium, you really don't have much tropomyosin moved out of the way. But as you get more and more calcium binding to troponin C, and then therefore tropomyosin moves, well, that facilitates other troponin Cs to accept calcium and further enhance the movement of tropomyosin. So you get more muscle contraction very rapidly as you start to reach a threshold of calcium concentration. And of course, that's very helpful for activating the muscle in a coordinated way. So you have this calcium spike that happens first transiently. Then you have calcium binding to troponin C, and that takes some time, so there is a delay. And then finally the muscle force that's created with myosin, the myosin head group binding to the actin and the whole process there. And so you can see that while the initial activation might be really fast here, within 10 milliseconds or so, the muscle force can last much longer, and there is a bit of a delay as well. All right, so then what happens with the skeletal muscle once you've got the tropomyosin moved out of the way and the myosin can bind to the actin? Well, the important components of this striated skeletal muscle include these myofibrils shown here in this upper diagram, which is a series of fibers that are put together as a rod, and then these rods are compacted together into a full muscle fiber. And I have to admit that sometimes I refer to a myofibril, other times I refer to a muscle fiber. Think of them as being pretty similar, but there is certainly a architecture within the muscle fiber to have these myofibrils. You have the T-tubules of the sarcolemma that we discussed as well. Those are these white dots shown on the surface of the sarcolemma that allow the depolarization of the muscle membrane to reach deep into the myofibril packets here. The sarcomere, consisting of thin filaments and thick filaments, as shown in this diagram here on the right center. And so let's go through this in a little bit of detail. The thin filaments only have actin, and there's no overlap with the thick filaments here. And so it looks like it's not a very dense area. These actin filaments are attached to the Z disc here, sometimes called the Z line. So they have an anchoring point to these actin filaments, and they're running to the left and to the right. And they're not attached on their end. That's not attached to the Z actin. They're just free on this other end. Similarly, the thick filaments are the myosin proteins, and the myosin is attached to a median line, sometimes called the M line. So it is permanently attached to a good base as well, and the other end of the myosin filaments are free to dangle, and then there's an integration, a meshing between the actin filaments and the myosin filaments, which is this dark band right here that you can see. And then there are other terms for A band and I band for parts of the myofibril that can absorb anisotropic and isotropic light. There's an H zone, and we've talked about M and Z. So there's a bunch of other terminology, but I just like to focus on the myosin and actin. I think it's also pretty cool that the thin filaments of actin and the thick filaments of myosin overlap with each other here in this AI overlap diagram. And the actin filaments are surrounded by a triangle of myosin, and a myosin protein is surrounded by 6 of these actin filaments. So there's really a nice geometrical orientation of these, and there's a reason for that we'll see on one of the next slides. Okay, so how does myosin and actin interact? Well, myosin consists of 2 heavy chains shown as these tails, a dimer of 2 myosins that are wound around each other. And then also these head groups that are attached to these heavy chains. Now there are 2 separate proteins shown in blue that are the light chains, 2 blue blobs. And we'll talk about that in the next lecture, in lecture 5. Really, we want to focus on these myosin head groups, and I'll refer to that quite often. They're actually oriented around the thick filament and are held together with titin, a very large protein. I think it's still the largest protein known in the human body. That's about 3.7 million kilodaltons, so a giant protein that holds the structure together. And importantly, the myosin head groups, if you were to look down the axis of the thick filament, are arranged where each of these head groups is rotated by 120 degrees. Well, as we saw in the previous slide, the actins, there are 6 of them around here. And so you can see that these head groups are interacting with different actin filaments that are hexagonally arranged around this myosin thick filament, right? So then actin is actually made of a protein called G-actin, a globular monomer, and when these polymerize together and form a helical dimer, shown as a bead of pearls, they're surrounded by tropomyosin, this other long fibrous protein, and troponin that binds to the tropomyosin. So then when the actin binding site for myosin is exposed, then these myosin head groups can attach to actin, and you've got interactions between those 2 fibers. Well, as you can see in this diagram, the myosin head groups are pointing towards the actin filament and binding. The actin filament, again, is as a base of the Z line. Myosin thick filaments are attached to the M band, the M line. So that's their base. And so then the thick filament of myosin and the thin filament of actin can slide back and forth against each other. And it turns out that's really the actin filament that's doing all the sliding, and the myosin really just stays in its place permanently. And that's how you get contraction. Somehow we're going to have these actin filaments slide to the right over on this part of the diagram relative to the myosin, or actin filaments will slide to the left over here in this part of the diagram relative to the myosin, and that will contract the fiber. And of course, if you have all of these different contractions happening throughout your entire multiple groups of thick and thin filaments, then you get major muscle contraction. Now, if you contract all the way so that the myosin is butting up against the Z line and the actin is butting up against the M band, well, you can't contract any further. You've pushed everything together. And in that case, you can generate no more force. So when you have the sarcomere length as minimal as possible, everything is crunched together as much as you can, well, there's no force that can be generated. And likewise, if you've expanded so that the end of the myosin is just barely touching the end of the actin, and you've pulled these fibers apart as much as possible to create the longest sarcomere length, well, then there's no force as well. It turns out that if you have about 2/3 of the thin filament and thick filament interacting with each other, that creates the greatest force, about 1/3 of the way from having the minimal sarcomere length. All right, so this cross bridge cycle is also very important for this part of this section of the chapter. And so I'm going to start with step 1, just like the book. And then we'll look at some videos. The videos start at step 3. I think it is a little bit nicer to think of it starting at step 3, but we'll just stay with the book and start at step 1. It's a cycle, so it doesn't really matter where you start. Okay, so before ATP enters this diagram, the myosin head group is attached to actin. The tropomyosin has been moved out of the place due to calcium binding. When ATP binds, that loosens up the binding of the myosin head group. It's changed conformation enough that it no longer fits within the binding site of F-actin, and it will then detach from actin. So in step 2 here, you see the detachment. In step 3, the ATP is hydrolyzed to ADP, an inorganic phosphate, and that changes the head group again, so it is a little more sticky. It has a little more affinity for the actin filament, so it'll bind again, and yet there could be movement during this part where the thick filament and the thin filament, myosin and actin, are not bound to each other. So it could bind to a new binding site on the actin. Then inorganic phosphate will then dissociate from the myosin head group, and that's shown in step 5. And that causes the head group to change conformation. And while it seems a little subtle in this diagram, it does change its angle attached to the actin by quite a lot. And that causes the actin to be pulled in one direction relative to the myosin. You can imagine sort of a finger going shoo, and pushing one fiber relative to another, right? Okay, well then the ATP dissociates, and that locks in the myosin head group onto the actin. It's a very tight binding at that stage. And then we go back to step 1, where the ATP again binds to the myosin head group, and that causes dissociation, and so on and so on. So this is a cycle. And so you get this walking of the myosin head group from one area of the actin filament to another, especially when this head group changes conformation. And at this stage in the lecture, we will watch this movie on YouTube. There are actually 2 movies that are complementary to each other. And I would encourage you to go to these YouTube videos and watch these how the whole cross-bridge cycle works as a dynamic movie. It's pretty cool. One other important component here is the costamere. This is the set of many proteins that links the muscle to the skeleton. So the muscle, thin filament and thick filament interact with each other. There's contraction in the sarcomere. That will cause the Z disc then, which is attached to these other ligands here, to interact with the costamere, then are attached to the extracellular matrix fibers that are outside the muscle cell. And these fibers could be in collagen, could be in perlecan, and the collagen and perlecan and tendons and ligaments, they are attached to the bone. So this causes your bone to move when you have muscle fibers contract. pulling on or then anchoring through this mechanism onto the bone. Now, an important protein to discuss is dystrophin. We've all heard of muscular dystrophy, which is really a series of many disorders for many of these proteins. Particular Duchenne's muscular dystrophy lacks dystrophin. I believe it's a complete lack of this protein due to a genetic mutation and cycling out of that protein then within the body. If you don't have the costamere components that are linking the muscle to the skeleton, well, you can still have some muscle contraction, but then it won't do anything because it won't move the skeleton. And eventually, if there's no use for the muscle, there'll be muscle wasting and a decrease in muscle strength over time. And of course, that's very deleterious to the patient's health. So that ends Lecture 4. I would like to point out that these figures in the section 3.5 and 3.6 were not used for this lecture. They're secondary, optional material. You can still read through them. They do relate to the text, but I don't think it's really essential for long-term understanding. So for the anatomy homework, I'm excited to discuss synovial joints, and we'll just briefly discuss this. The homework does a great job of discussing 6 different mechanisms. There are 3 shown here, 3 on the next slide. There's a plane joint where 2 parts of your body slide again next to each other. That happens in your wrist. It happens in midfoot as well. You may not think that there's a lot of motion, but there is enough that you can kind of move your hand back and forth without moving your arm, and that helps to ensure that you don't get injured. A more interesting joint is the pivot joint. I love this idea that at the top of your spine, you've got a little post here, and your skull rests on that post, and your head can pivot to the left and to the right. Now, this doesn't control your head going up and down as if you are nodding. That's controlled by the spine itself. Doesn't control your head nodding back and forth. That's also a spine motion. But if you just want to look at rotation of your head and swiveling on a pivot, that's pretty cool, right? Also, there's a pivot joint in your elbow so that your arm can twist to the left or to the right as well. The hinge joint is pretty obvious. The knee or the elbow are an obvious example of a hinge. You only want your lower leg to swing forward or backward relative to your upper leg. You don't want it to go side to side. We have ellipsoid joints, not too many in the body. A classic example is in the wrist, where your hand can rotate around as if it's in the joint is in a shallow pan, an ellipse, and so you got a lot of motion there. Saddle joint, there's one in your thumb that's good to talk about. Not way down here, but right here, kind of at the base knuckle. And so your thumb goes back and forth in this motion. It's not supposed to go to left or right or bend your thumb, right? That looks like it would be pretty painful. Now down here, there's extra things you can do with your thumb, right? So I'm not talking about down here. It's more here where you got a saddle joint. And then you have a ball and socket joint in your shoulder, and that allows rotation in all 180 degrees around that ball and socket. And I guess maybe even more motion, right? I can go up and down here as well. Now, if you want to see extra detail of these main joints, well, here's detail. I would go through this in detail. It is part of the homework. And then here's a blow-up of each of these individual joints, but these are extra slides that are kind of repetitive. That brings us to the end of Lecture 4. Now, of course, in class, we kind of have more of a discussion. I hope this relatively short video is still very helpful for you. Thanks for watching.